Hello, welcome to Watch Creative Proteomics video. Today, we are going to introduce the phosphorylation analysis by mass spectrometry. Protein phosphorylation, a reversible process, is characterized by adding phosphate donated from ATP and removing phosphate from a phosphorylated protein substrate. A process is catalyzed by protein kinase and phosphatase. In eukaryotic cells, about 86.4% of the protein phosphorylation events occur on serine, while 11.8% protein phosphorylation events occur on threonine, whereas only 1.8% on tyrosine residues. Protein phosphorylation plays important roles in many cellular processes, including cell cycle, growth, apoptosis, and signal transduction pathways. In addition, phosphorylation can modulate enzyme activity, alter its affinity to other proteins, and transmit signals through kinase cascades that often are branched and interactive. In order to understand these mechanisms, it is necessary to analyze protein phosphorylation. There are many methods for phosphorylation analysis, including kinase activity assays, phosphor-specific antibody development, Western blot, enzyme-linked immunosorbent assay, as well as intracellular flow cytometry and immunocytochemistry or immunohistochemistry. In addition to these methods, mass spectrometry analysis plays an important role in phosphorylation analysis. There are two common strategies for identifying protein phosphorylation sites: by bottom-up proteomics, including identifying phosphorylation sites from individual proteins and small protein complexes, and identifying global phosphorylation sites from the whole cell and tissue extracts. Identifying phosphorylation sites of a specific protein of interest is important for comprehensively study the functional role of phosphorylation from the protein. There are several steps, including sample preparation, analysis by tandem mass spectrometry, and data analysis. For sample preparation, because of the low stoichiometric level of phosphorylated peptides compared to unmodified peptides, there is a need to purify as much protein as possible. In addition, to enrich phosphorylated peptides, there are several methods available, including metal ion affinity chromatography and titanium dioxide beads. For mass spectrometry analysis, the peptides can be introduced into the mass spectrometer through liquid chromatography coupled to electrospray ionization, or matrix-assisted laser desorption ionization. Peptides are most commonly sequenced by tandem mass spectrometry using collision-induced dissociation. Electron capture dissociation and electron transfer dissociation can also be applied for phosphorylation analysis. And informatics software is necessary to identify peptide sequences and their phosphorylated counterparts. The advent of methods for enrichment of phosphoproteins in conjunction with improvements in mass spectrometry has resulted in the identification of large numbers of phosphoproteins and phosphosites. From hundreds of studies, more than 300,000 phosphorylation sites have been identified, across a multitude of both prokaryotic and eukaryotic species, with more than 200,000 of these coming from mammals alone. Greater than 95% of these sites have been identified using mass spectrometry-based phosphoproteomics. With the advancement of mass spectrometer that allows for the rapid and sensitive sequencing of peptides, and the concurrent development of phosphopeptide enrichment strategies, we now have the ability to analyze changes in protein phosphorylation at an unprecedented scale. Global phosphorylation site analyses that capture phosphorylated sites from biological sources. Sites are more time-consuming. It has several strategies. The first strategy involves digesting the whole cell lysate, followed by peptide fractionation by strong cation exchange chromatography, and phosphopeptide enrichment by metal ion affinity chromatography and titanium dioxide beads. In the second strategy, the protein lysate can be fractionated by SDS-PAGE, followed by digestion, phosphopeptide enrichment, and liquid chromatography tandem mass spectrometry analysis. In addition, one can also immunopurified only phosphotyrosine peptides using a phosphothreonine antibody followed by liquid chromatography tandem mass spectrometry analysis. Creative Proteomics has established a highly sensitive HPLC tandem mass spectrometry platform that can analyze phosphorylation in multiple samples, and in both eukaryotic and prokaryotic organisms. In addition, we have optimized our protocol to enable more fast and sensitive site mapping service for phosphorylation analysis. Thanks for watching our video. At Creative Proteomics, we provide the most reliable services. If you have any questions or specific requirements, please do not hesitate to contact us. We are very glad to cooperate with you.